Hello everyone, my name is Xiaotong Liu. I'm a graduate student in bioinformatics and computational biology at the University of Minnesota. My topic today is multi-compartment models, a mechanistic modeling approach for interpreting transcriptome dynamics during plant immunity. Let's begin with some introduction of plant immunity. So the plant doesn't have adaptive immunity like humans or mammals. It can only rely on its innate immunity to defend themselves. There are two major modes of immunity, including pattern-triggered immunity and effector-triggered immunity. So when pathogen attacks a plant, the plant can recognize the pathogen with its receptors and co-receptors on the cellular surface. And after signal transduction, a series of PTI response will be triggered. Some pathogens can escape from PTI response by delivering the effectors into the plant. And the plant have also developed another phase of response, which is ETI response, to detect the effectors and effectively defend themselves. So here, there are two types of effectors, including AVR-RPT2 and AVR-RPM1. In my study, I mostly focus on the first one, AVR-RPT2. Studies have found massive transcriptional reprogramming during plant immunity in Arabidopsis. In this publication, Ma, Akira et al., the transcriptomes were profiled at 8 time points upon infection with the virulent bacterial strain DC3000 and 2 avirulent strains expressing AVR-RPT2 or AVR-RPM1 plus water as the mock. In this figure panel, they plotted the number of differentially expressed genes at different time points. We see that before 3 hours, there are very few genes that are either upregulated or downregulated. But after 3 or 4 hours, thousands of genes are significantly upregulated or downregulated in all these 3 conditions as compared to mock treatment. People can use the heat maps to show the expression pattern difference between these 3 conditions at 8 individual time points. And in this study, we want to develop a mechanistic approach that can model the temporal dependency in the data and summarize the gene dynamics in a more compact way. We developed multi-compartment model for this. There are 2 steps for constructing multi-compartment models. The first one is to generate a chain. So we firstly define a source node following the exponential decay. And we add one more compartment to this, which is linearly activated by the source node, and there is a first-order decay with this compartment. Based on the same idea, we can have more compartments until we have 6 compartments after the source node. So we may characterize the dynamics of these inputs with such curves, and we see that the compartments that is closer to the source node has relatively early peak time, and vice versa. The second step is to feed the temporal transcript data with a 2-input combination. So we collect the transcript data for each gene. We may select the best 2-input combination for this gene. And we, based on the same idea, have a differential equation. And when we assume negative binomial distribution of the count data and with maximum likelihood estimation, we can characterize the parameters within this differential equation and use these parameters to further interpret its dynamics. We have evaluated the goodness of fit of the genes. Here, 4 example genes are plotted, and we see the goodness of fit is pretty decent. We can also detect ETI effects with our eyes. We see the top genes there's a clear early peak in the AVR_2 condition, and there's no early peak in the DC3000 condition. We can also see some late ETI effects in the second gene here. We can use the heat map to show the transcriptome-wide ETI effect. More than 93% of the genes have a larger early peak in the AVR_2 condition as compared to the DC3000 condition. We tried explaining the early ETI regulation with transcription factor binding data. We selected more than 300 genes with significant early ETI effect as positive genes, and selected the same number of genes with no significant expression difference from MOC at all time points as negative genes. And for each transcription factor, we basically count the number of binding events within the promoter region across all the positive and negative genes. And we did this with 349 transcription factors. We applied random forest for prediction, and we get around 0.7 accuracy rate. So here I listed several important features or transcription factors for the prediction. It includes worky transcription factors which have been confirmed to involve in the ETI effect. I want to thank my advisors Chad Myers and Fumi Kataguri for their suggestions and feedbacks for me in this project, and we're also very grateful for the funding support from NSF. Thank you for watching.